Right, so up to now what we have learned, we just talked about there was severe injury, chemical mediators of inflammation were produced, arterial dilatation, increased blood flow, then microcirculation increases its permeability, so protein-rich fluid exudate out, and due to that reason, hemoconcentration in microcirculation, increased viscosity, stresses, laminar blood flow is disrupted, right? RBCs clump together, marginate the white blood cells. This is the onset of cellular event. First of all, due to hemodynamic changes, there is margination of white blood cells. Then we talked about that selectins on one cell will act with sialylated sugars on other cell, and there will be adhesion activity, loose adhesion between the endothelial cell and white blood cells, right? And that will start the process of rolling. Again, let me tell you that P-selectins are expressed by endothelial cell when they are stimulated by histamine, thrombin, and leukotrienes. And E-selectins are expressed by endothelial cells when endothelial cells are stimulated by tumor necrosis factor and interleukin 1. Another special type of selectin is sometimes expressed on white blood cell, that is called leukocyte selectin or L-selectins. Right? What really happens, selectins either expressed on endothelial cells or expressed on the white blood cells, they interact with sialylated sugars or on the other complementary cell. Normally, here we have shown the P-selectins and E-selectins, they will interact with the sialylated sugars which are normally expressed on leukocytes, extensions, right? And when leukocytes interact with P-selectins and E-selectins, they are loosely attached, and under the hemodynamic pressure, they keep on rolling, right? So, selectins sugar interaction is a loose interaction, but meanwhile, chemokines are produced. Chemokines reach to the endothelial cell. The endothelial cells have special protein which hold the chemokines, and chemokines which are sticking to the endothelial cell, they do a special stimulation to rolling, special stimulation to the rolling leukocyte, so that their integrins with low avidity, they switch to a new conformational change which is called high avidity, right? Meanwhile, when integrins are in full action, they are presented with ICAM and VCAM. Integrins interact with the ICAM and VCAM. This is a very strong adhesion, so now leukocytes don't roll anymore and they stick to this endothelial cell. And we say when lot of white blood cells are sticking to the endothelial cell, we say that endothelial lining has developed a pavement made of white blood cells. Is that right? Now, what we have to see, that purpose of holding the white blood cell in this endothelial lining was to get them out towards the focus of injury. Now, next point which we have to see is that how white blood cells get out of this area into interstitial area, and how they eventually move towards the focus of injury, and eventually they they attach and engulf and destroy the bacteria or injurious agent. So let's study step by step. The next step to study is how the white blood cell get out of vascular compartment to extravascular compartment. The movement of white blood cell from vascular compartment to extravascular compartment is called immigration, or it is called extravasation, or it is called diapedesis. There are three names used for this, so you should not be confused. The process by which white blood cells from the vascular compartment come to the extravascular compartment, there are three terms. We say they are migration, you can say. They are immigrating from vascular compartment to extravascular compartment, or we call it, yes, extravasation, getting out of vascular system. Or another term which is used for that is diapedesis. So these all are one and the same thing, right? That white blood cells are immigrating, or they are extravasating, or they are showing the phenomenon of diapedesis. Now, how the white blood cells really move from here out? Because let me tell you, up to now we have seen that one type of adhesion molecule stick with the other type of adhesion molecule. Now what happens, that when white blood cells are present sticking to the endothelial cell, meanwhile another class of some more cytokines are going to the white blood cell and also acting on endothelial cell. Is that right? More cytokines, they are going to the acting on endothelial cells and white blood cells, right? Now, the special type of influence expresses a special type of molecules on white blood cells. And these molecular motors, right, look at it. They are, the molecules now which are expressed by endothelial cells and expressed by white blood cells are same, right? What is this? This is what was least expected. It is HOMO. The both molecules are same. Don't laugh, it is really HOMO. The molecules from both sides are same, right? In the books you will find books like so respected books like Robbins, they write homophilic attitude, homophilic molecules, right? The molecules get HOMO, the type of molecule which is expressed by endothelial cell, same type of molecule is expressed by the leukocyte, and they start rubbing each other. And when they start rubbing with each other, they act like molecular motors, right? And what really happens then? White blood cell starts sticking down. Let me show one white blood cell properly transferring from here. Should I make a really good diagram for you? You deserve a good diagram, I think. Look, okay. This is one endothelial cell with its nucleus, of course. One endothelial cells. Now, white blood cell was sleeping here. But not sleeping, it's really resting there. Okay. Now what happened? More cytokines come, they act on both sides. This time, what really happened, the molecules which are expressed by they are same or different type? They are? Now these molecules interact with each other, and these molecules, when interacting with each other, white blood cells start sticking down. And it's not only from one side, actually it occurs on from both endothelial cells. Is that right? So molecular cells which are coming from the endothelial cells, adjacent endothelial cells, they start act on the similar molecule on white cells and start gripping it like this. Now the white blood cell starts slipping and squeezing itself in between the endothelial cells, right? So what you find after some time? What you really find after some time, okay, I will make the next stage in the next diagram, that white blood cell and of course, what is this? The interaction of what type of molecules? Homophilic interactions. Homo mean, all of you know that. Philic mean love. Homophilic, molecular love, right? Similar molecular loving each other, and the result is bad, it will lose its position. Right? Because it's going out. It was very happy with the endothelial cell, but now it's getting out. It is being squeezed out. And what is this? This is basement membrane, right? So as time will pass by, eventually you will find the white blood cell has come over here. Is that right? So again, what pushed the white blood cells while they was sticking or they are having good adhesion with the endothelial lining to the extravasation? The process is homophilic molecules. I think we should give them a name. That will be more respectful. They put a case against you maybe. So these molecules are called funny name. They are called PCAMs. PCAMs. PCAMs mean what? Yes. It reminds you something. Right? PCAM. PCAM stands for what? Platelet. Yes. Not anything else. Platelet. What is this? Platelet cells adhesion molecules. Originally they were discovered in platelets, but later on they were found that same molecules help the white blood cells to squeeze through the interendothelial gaps and help in extravasation, right? when they come over here, white blood cells really are exhausted. I mean, it was not easy interaction, isn't it? So they are very, very exhausted. So what they will do? They will rest. They have come out of endothelial lining, but still within the basement membrane. Is that right? They are very exhausted now. They rest for a while, and after that, what they happen? A lot of cytokines still stick to them. They become very active, and they start releasing very, very dangerous, you can say, what? Enzymes. What enzymes they are releasing now? These enzymes are called collagenases, because basement membrane is made of collagen. What type of collagen is present in basement membranes of? Yeah, many basement membranes have type 4 collagen, especially glomerular basement membrane has type 4 collagen. For this you will read and tell me, right? So what really happens, that these neutrophils, they start releasing, or white blood cells start releasing collagenases, and these collagenases digest away the collagen in this area and produce a hole there. And through this hole, you will find after some time, the white blood cells have come out. And a little more moments, you will find a lot of white blood cells which have come out of microcirculation. So one microcirculation, this is another microcirculation, right? And there are also other microcirculations. So from now this process, in which from the vasocompartment, white blood cells have shifted to extra vasocompartment, this is called immigration process or extravasation process or diapetesis process. The molecules were involved was PCAMs, right? And these PCAMs are also called CD31. These molecules CD31. Why they call these molecules CD31? Because they were expressed, you know, CD means cluster designation. On some cluster of the cells, these molecules were discovered in sequence number 31, right? Actually, what happened when we discover a new protein, we just say cluster designation 31, 56, whatever. Later on, we find what this molecule is doing. So when this was discovered first time, this molecule, we call it just cluster designation protein number 31. And now we know this is actually a protein which acts as a motor molecules to help the white blood cell to extravasate. Am I clear? No problem up to this? So white blood cells are here. Now next question is, white blood cells have come out of vascular system, right? They should move towards the focus of injury. It should not happen that this white blood cell should move in this direction. It should not move in that direction and go somewhere else and miss the point where the action is required, isn't it? It should really move. This army of these white blood cells which has just immigrated must move towards the focus of injury, where there is, let's suppose, many bacteria and local cells, and there are injured cells and bacteria multiplying, a lot of chemical. Now the question is that how white blood cells move in this particular direction? Yes, who tell the white blood cells how to move in a particular direction? How the white blood cells sense that in which direction they should have the locomotion? Yes. Actually, yes, they are chemical mediators. Look, at the site of inflammation, the concentration of chemical mediator is maximum. As you move away, chemical mediators concentration become less. So what really happens, that white blood cells can sense the concentration of chemical mediators, and under the concentration gradient of chemical mediators, they display a directional movement. And this directional movement of white blood cells under the influence of chemoattractant molecule is called chemotaxis. 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 What is chemotaxis? Chemotaxis is directional taxi of the cells, movement of the cell, right? Direction by what? Chemical substance. You know, as you people sometimes display, when you are very hungry, you also show some type of chemotaxis. When you feel there is some good food being cooked and you start moving, is that right? That is also a type of chemotaxis. There are chemical substances from good food, you know, chemical substances which are volatile, they come here as good smell, and then you can smell the food and move in a particular direction. That is positive chemotaxis. But if there is some bad smell somewhere, usually you move away or towards? What about you? You still move away from bad smells. Good. So if someone moves away from the bad smell, we say negative chemotaxis. Right? In the same way, white blood cells are attracted by the chemical substances which are produced at the focus of injury. Right? These are many chemical mediators and different chemical substances. Some chemical substances are products of bacteria. Some chemical substances are products from our cells. For example, at this side, bacteria, especially bacteria, produce special type of chemical substances which attract the neutrophils. Bacteria produce special type of proteins or peptides which are having at the end N-formyl methionine amino acid. Bacteria produce special type of protein molecule which have a special type of amino acid at the end of the protein. The amino acid is called methionine, and methionine is having a formyl group. So N-formyl methionine amino acid at the end of certain peptides which are produced by bacteria, and our neutrophil can smell those molecules and move after them. I'll tell you how they move. They don't have nose, right? So in the same way, our injured cell also produce some chemoattractant. That special call to neutrophil, please come here, we are in trouble. For example, injured cell can also produce leukotrienes, especially leukotrienes B4, right? Leukotriene B4 are produced by the injured cell membranes of mast cells, macrophages, or any other cell, and they are very strong chemoattractant of what? Neutrophils. Leukotriene B4. Don't call it leukotriene, because in the books it is written leukotriene. We can call them leukotrienes because they produce the trains of the white cells coming to this direction, right? Leukotrienes B4 or leukotrienes, right? The right word is leukotriene or leukotriene? Right word is leukotriene, but they produce leukotrienes, right? Leukotrienes mean trains of the white blood cells. Is that right? You are not understanding it. No problem. Look. No. I have to get you explained. Right, listen. What is there? There's a lot of injury. Injured cell membranes release prostaglandins and leukotrienes. Is that right? One of the leukotriene is leukotriene B4. There are special receptor for leukotriene B4 on the special sensors of leukotriene B4 on the leukocyte, and leukocyte move towards the high concentration of leukotriene B4. If leukocytes from all sides are moving towards this, one leukocyte going, then another going, then another going, so leukotrienes are producing the trains of leukocytes. You know, train means many leukocytes one after the other, moving toward the site where the battlefield is there. Is that right? Now I'm clear? In the same way, if you know that lot of plasma proteins which leak out, some of them are complement factors. Some complement factors are activated like C5a. You will study this in complement lecture. So plasma protein which come here, one of the protein is complement factor which is broken down into C5a component. Neutrophil can also sense this molecule and move into that direction. So what really happens? That at the site of injury, there may be some special substances which attract the white cells. These chemoattractants are called chemotactic agents. What are they called? Chemotactic agent. Some chemotactic agents are produced by external factors, like bacteria produce chemotactic agent, right? These chemotactic agents which are produced by bacteria, they are called exogenous chemotactic agent. And some chemotactic agents are produced at the site of injury by our own body. For example, injured cell membranes produce leukotriene B4. Like plasma proteins may produce C5a, right? Then these are because produced within our body, so they are called endogenous chemotactic agents. Again, what is chemotactic agent? Chemotactic agent is a molecule which attract the white blood cells at the focus of injury. Some chemotactic agents are exogenous, others are endogenous. Am I clear? Now the question is that how the neutrophil knows in which direction to move. Neutrophil does not have eyes to look where are the chemotactic agent, and neutrophil does not have a nose to smell where are the chemotactic agent. Then how a neutrophil really knows how to move in a particular direction? Let me explain this. Look, this is a neutrophil. I put one neutrophil here and I put one neutrophil in this corner. Now this has to move in this direction, this should move into that direction. How it really happens? Actually, let's suppose chemotactic agent is leukotriene B4. Now all of them, neutrophil has, white blood cell has receptors for leukotriene on all over the surface. It has receptors for leukotriene special proteins which can sense the presence of leukotriene all over the surface. But leukotriene is more in this area, and as you move outward it becomes less. Now tell me, leukotrienes will bind more on these receptors or leukotriene will bind more on back receptors? In the front. So leukotriene will bind here, but not there. It will bind more on this side and this side, but not there. So what really happens, then they give signal to the cell. Their chemical mediator is on this side. And I will tell you how the molecular motors are activated and the cells start moving into that way. First, just take an example. If I'm a white blood cell, this is a blood vessel, I've just come out of the vessel. I've closed my eyes because white blood cell does not have eyes. Nose, I don't want to close. Now, like a white blood cell, I'm standing. On my skin all over, there are receptors for leukotriene B4. If there is injury there, it means leukotriene is produced by there. So it will bind on my right arm more or left arm more? Right. So when this will touch the receptor on this side, this will move like this. You get it? If it start moving, putting there, it will start moving like this. If it start touching on the back, then it start moving like that. So what really happens? That neutrophil, you put it in the center of a container, put a drop of leukotrienes on the other side, it will start moving to that side. So you understand it? That leukocytes have receptors all over the body. The receptors are just like buttons. You touch the button of any side, it start moving to that. Now question is that how it really moves, I will tell you. Because they don't have feet even, isn't it? But they do make feet, micro feet. So now you understand that they have receptors all over, and which side on the cell membrane, that part of the cell membrane where receptors are activated, the special type of foot processes are made and it starts slipping there, right? Let me explain also something interesting. Let's suppose, now I make one leukocyte large. What really happens? Let's suppose this is a receptor for the chemotactic agent. What is it? Receptor for chemotactic agent. Let's suppose this is a receptor for leukotriene B4 or interleukin 8, whatever. Now, do you know what is the type of this receptor? All of you know, almost all of. Now, leukotriene bind here. When this receptor is stimulated, look at this receptor. What is this type of receptor? 7-pass receptor. All of you have studied 7-pass receptors. They are coupled with intracellular which proteins? G protein. It is coupled with GQ protein. When this receptor is stimulated by chemotactic agent, they stimulate the GQ protein. GQ protein loses the ADP and loses the GDP and acquire very bad GTP. These are G proteins. They are driven by the guanosine molecules. I will not go into that detail. That you will do in the receptor. For a while you just understand different chemotactic agent bind with 7-pass receptors. Receptor activate, let us suppose in this example it activate GQ protein. GQ protein will stimulate a special type of protein here. Right, this is a very, very special type of protein in the cell membrane, and this protein is a dangerous enzyme. What is it? It is dangerous enzyme, and this protein is called phospholipase. Okay, let me make it more clear. Phospholipase C. Now GQ will stimulate on the back of phospholipase C. Phospholipase C activate and start digesting some phospholipids in the membrane. And there are special type of lipids in the membrane. What are the name of those lipids? Phosphatidyl inositol diphosphate. So phospholipases are present already in the membrane. So phospholipases break down the phosphatidyl inositol diphosphate into diacyl glycerol and inositol triphosphate. Now again listen, it's not that difficult. It's just like this. You go to someone home, push the button. What is that button? Doorbell, I mean. You push that button and they start ring inside the home. But just pushing the button and train, there's something in between, lot of mechanical activity. In the same way, this is pushing the button and there's intracellular signaling going on. So what happens, your finger is chemotactic agent, this button is 7-pass receptor, which is coupled with G protein. This G protein, GQ protein stimulate phospholipase C. Phospholipase C break down the lipids of the membrane into diacylglycerol and IP3, right? IP3 is inositol triphosphate. Within the cell, there is what? Endoplasmic reticulum. Endoplasmic reticulum has a lot of stores of calcium. Very good. Excellent. Endoplasmic reticulum store of calcium. Actually, there are special type of IP3 sensitive pump here. What are these? Calcium pump which are driven by IP3. So when in the cell, inositol triphosphate level go high, they stimulate these pumps and these pumps start throwing out calcium into cytoplasm. Calcium will bind maybe with calmodulin, and also stimulate protein kinase C. Are you understanding what's happening? You just touch the chemotactic agent, whole chemical reaction start within the cell. Signaling mechanisms are activated. Is that right? Now, this calcium and calmodulin with protein kinase C, they start phosphorylating certain proteins. End result is that, you know what happens? In the cell, the special type of these molecules. What are these molecules? These are actin molecules. What are these molecules? Actin molecule. What really happens, the calcium and other signals force the actin molecule to polymerize. So under this receptor where all this reaction occur, actin molecule start sticking with each other and they start making a chain. Many actin molecules, actin monomers fuse with each other and make actin polymers. So special tubes, special type of actin filaments are made in the cell. Is that right? Which part of the cell? Where chemotactic agent is working. These actin filaments will not form here. Again, let me tell you. If this is the white cell, you stimulate here, actin filament will form here. If you stimulate here, actin filament will form here. If you stimulate here, actin filament will form here. You are understanding? How the actin filaments are formed? That when all these signaling mechanisms result into polymerization of monomers of actin into polymers of actin, and they start making filaments. You are getting it? Filaments are being formed in a funny way. You know how they form? That new monomers are added on this side, and monomers from the back are breaking away. Monomers on the back are breaking away. Let me show you here. That suppose these are actin monomers, actin chain. Now these are two actin chain. Now listen. Where the receptor is activated, new actin molecules are made on this side. Right? It means filaments are growing in this direction. And from the back, they are breaking away. They are breaking away. So what really happens, that in the end, they are breaking away. And then, so cell has lot of actin monomers, they are constantly adding in the front, is that right? And breaking away from the, you say this is the leading end and this is the trailing end. From the trailing end, they keep on breaking and they keep on adding on the leading end. So what will happen? These filaments will grow and they will push the membrane forward. They push the membrane forward. So you will find the cell become like this, because what is being formed here? Actin filaments. With that, there will be some also myosin filament. They start interacting, right? And start. Now, as it keep on moving more and more forward, this will be pushed more forward. And when they are pushed more forward, from this side it will be pulled, right? Now from here it is disassembled, it has reached there. So what really happens? Have you ever played with that thing? Have you seen the front wheel cars? What happened? The front wheels are moving and dragging the rest of the car with it. I think all of you must know there's something called front wheel cars. It does not mean that wheel should be in the front of the car. You know, front wheels are moving. Actually, your white blood cells behave like front wheel cars. right? What are the front wheels? The wheels are actin filaments, and these actin filaments are constantly growing in the direction where the membrane is stimulated by chemotactic agent. So that part of the membrane bulges forward. That forward bulge of the membrane is called, the special name, that name is called philopodia. This, let's pose it here, philopodia or podium if there's one. So what really happens, look, here receptors are stimulated on this side, so this will develop front wheel on the molecular front wheel on this side, this will develop on this side, this will develop on that side, this will develop on this side, and you get it? Now again, if focus of injury is here, and this is your microcirculations, I'm making a small diagram. White blood cells have come here out, here out, here out. It will develop front wheels into this direction, molecule on this direction, and so all of them will start moving to the focus of injury. Again, let me repeat it. Focus of injury is releasing chemical attractive substances, chemoattractant like C5a complement or leukotrienes B4 or interleukin 8 or bacterial product. All these things stimulate the, what is this? White blood cell receptors. That part of the membrane of the white blood cell where receptors are stimulated. What are these receptors? 7-pass G protein coupled receptor, right? Which act through GQ, activate phospholipase C, which increases intracellular level of IP3, calcium comes out, and calcium colmodulin and protein kinase C is activated. Many proteins are phosphorylated. End result is that under the whole under that part of the membrane where a lot of signaling is going on and proteins are phosphorylating, actin filaments are being formed. And actin filaments have a very unique feature. For example, my fingers are actin filaments. That if membrane is stimulated on this side, then my fingers are growing into that direction. On the back they are dissolving and again growing on that direction. So what they will do? They will keep on pushing the membrane forward, and rest of the white cell will be dragged with that, just like front wheel car, right? And moving the front wheels, and rest of the white cell is moving after that. In this way, white blood cell show a directional movement towards the focus of the injury under the influence of the chemotactic agent, and whole process is called chemotaxis. What is it called? Chemotaxis. Am I clear? Have you ever played with the sack race? You put a sack and then just try to move. I think that's the better example. You know, neutrophils are like sack race. That neutrophil standing like this, sac is its membrane, right? And anywhere you touch, it moves its feet on that side and start moving like this. Is that right? In this way, neutrophils start moving towards the site of injury. Meanwhile, we don't want it, they overshoot and run away. What really happens? As neutrophils are coming here, on the way, they develop special type of integrin molecule into full action. Those integrin molecules are different than other integrin molecules. Those integrin molecules bind with extracellular CD44 molecule. As neutrophil is moving to this direction, they express special type of, what is this? Beta-1 integrins. And there are special type of these molecules which are called CD44. They are present throughout the interstitial fluid, such molecules. And what are these? These are the footstones for the white blood cells. And these footstones are present in all the interstitium of your body. What are these molecules? CD44. Now, so white blood cell stuck their foot on this so that they stand over there strongly. So while they are coming for the war, eventually on these molecules they stand and they make a whole warring zone here. Advantage of this is that they will be not only through inflammatory process, white blood cells have been delivered to the site of an injury and a site of inflammation, but they are also held tightly there so that they will do what? Yeah, they will start doing, they will undergo full activation, and then they will start doing the phagocytic process, right? That we'll discuss in the next lecture. How white blood cells, once they come over here, they become fully active. Like they show the muscle before the fight. That muscle showing and all that activity is simply called white cell activation. And after that, they start phagocytosis. We'll talk about that in the next lecture. Is there any question? Okay, few clinical point I would like to highlight. Number one, sometimes white blood cells don't have, and sometimes white blood cells don't have the sugar molecule. They are not sugary. Do you think they will stick well and go out?
No.
So it's not good to be without sugar. So sometimes some white blood cells don't have sialylated sugars expressed, right? Because the genes which make special enzymes so that those sugars will be available, those genes are not working well. If those genes are not working well in a person, then what will happen? The white blood cells will not make those enzymes, they will not express those enzymes which can help in the proper expression of what? Sialylated sugars. And if white blood cells are I should say sugarless or sugar-free? Sugarless. So when they are sugarless, they are not sweet cells. Do you think these hookers will be interested in them? Even they have their taste, you know. So what really happens, endothelial cells do express their, what is this? Selectins, right? But these selectins are unable to find the sugars on, sialylated sugars on white blood cells. So what really happens in these patients, that during inflammatory process, white blood cells do come on the margin. They do bump there, but they don't get really under sticky and rolling. And if there's no rolling and they bump back, then of course there's no activation of integrins and immigration. So at the site of inflammation, chances of getting white blood cell is more or less? Less. It's good news or bad news? It's a bad news, isn't it? So it's not good to be without sugar. Sometimes you should respond to certain signals. That's a normal capability. Is that right? Right. So what is the meaning of lad? Who has good English? What is lad? Lad. Lad means boy, isn't it? Guy. Right? Do you think a lad which is not sugary and not interested in this type of seductive activities, he can be number one lad? He is called lad 2. This disease in which white blood cells do not have sialylated sugars on their surface and they are unable to, white blood cells or neutrophils are unable to interact with the selectins and they are unable to roll properly, we say the person is having a disease called lad 2. This lad does not mean really boy or guy. Lad is standing for leukocyte adhesion deficiency. What is this? Leukocyte adhesion deficiency. So you can imagine sometimes there are very, you can say, what I should say, indifferent type of neutrophils. They are not sweet neutrophils, right? They are not having sialylated sugars. So they don't interact with the endothelial cells expressing endothelial cells which are expressing selectins, right? And these neutrophils, of course, don't help much in the inflammation. Is that right? This condition is called leukocyte adhesion deficiency disease number 2. This is disease number 1. We'll talk sometimes else. Another thing which I would like to highlight is, okay, actually what happens that some people who have high levels of catecholamine or corticosteroid, or they have been given lithium as a drug. Again, there are 3 conditions I've said. People who have in their blood high level of catecholamine, what are catecholamines? Epinephrine, norepinephrine and dopamine. They are called catecholamines, right? Secondly, corticosteroid, they can be given as drug also, corticosteroids. And third drug which I've mentioned is lithium. Lithium is given, these are not lithium batteries. The lithium bicarbonate tablets, they are given for what? There is a disease called bipolar disorder, right? In which person especially get sometimes very pathologically happy, we call it mania, or very depressed. So person mood remain either at the depressed pole or become very happy pole. Probably the mood regulators in the brain are disturbed, yes. And lithium ion somehow regulate your mood regulator and the mood becomes stabilized. They have found the people who have high levels of catecholamine in the blood or high level of corticosteroid in the blood or high level of lithium in the blood, in these people, if you take blood sample from a vein, neutrophil level is high. We say these patients have neutrophilia or neutrophilic leukocytosis. The neutrophil count in the blood sample is high. Who will explain why? Anyone? Patient or persons who have high levels of circulating catecholamines or high levels of glucocorticoids, or they are taking lithium drug, right? If you check their, take a venous sample of the blood, you will find neutrophil level high. What could be the reason for that? No, this is a very simple reason. Actually, all the time, some of the neutrophils are sticking loosely with your endothelial lining. Even when where there is no inflammation, very few neutrophils are all the time sticking with the endothelium. You are understanding me? Now, actually, catecholamine, corticosteroid and lithium do not allow the endothelial cell to express the adhesion molecules. That is why catecholamines or corticosteroids or lithium, they inhibit the capacity of endothelial cell to express adhesion molecule. So it means the neutrophil will adhere, they will stick more to endothelial cell or they will stick less to endothelial cell? Less. And if they are not sticky to endothelial cells, they are in main circulation. So in the circulation, apparently it will look from the blood sample as if neutrophil count is high, but total body neutrophil count may not be high. Am I clear? Opposite to that, endotoxins. Endotoxins are the substances which are released by especially gram-negative bacteria. If your blood has a lot of endotoxins, endotoxins help the endothelial cell to overexpress what? Adhesion molecules. Endotoxins act on the what? Endothelial cell, and endothelial cell express too much adhesion molecule. So if I have endotoxemia, if there are too much endotoxins in my blood, my endothelium is activated. Most of the neutrophil will be sticking with the endothelium. You take my blood sample, neutrophil will be coming less in the blood. So there will be neutropenia. So, but of course, neutrophil count in the blood sample will be less, but truly in the body, neutrophil count will be normal. So it's an interesting phenomenon that there are some conditions which reduce the neutrophils sticking to the endothelial lining and increase the neutrophil count in the blood sample, like catecholamine, corticosteroid and lithium. And there are conditions like endotoxemias which activate the endothelial cell too much. Endothelial cell all over the body express adhesion molecules, and most of the neutrophil get sticky to the endothelial cells, and when you take blood sample, neutrophil count will be high or low? That will be low, and that will be as if there is neutropenia. Here we stop our lecture and we'll continue rest of the discussion in next session. Class dismissed.